Everybody on a very pleasant summer evening, and particular welcome as somebody's just pointed out to me that I know absolutely nothing about sport because apparently there's a football game we're competing with. So thank you very much for coming along to this. This is our fourth departmental event for alumni, and it's the second one that we've had early evening in London to complement the meetings in Oxford as well. So I'm delighted to see you all here. I looked at my crib sheet and I was pleased and surprised to see I'm going to talk to you about the departmental status. And that sounded like a rigorous statistical survey could be a tad on the dry side. So I thought I'd just go through a few brief highlights and then let my colleagues talk about more exciting matters. So it has been a good year. Of course I have to say that. The head of the department has to smile on all occasions, but I think it's been an excellent year both for research and teaching. And I think that's particularly important and gratifying during the slightly difficult times that universities in some ways are facing at the moment. In terms of research, the immense efforts of all my colleagues have been rewarded with research grants from a whole range of different funds from government sources, so that's both the research councils within the UK and also increasingly from the ERC as one of the major European funders of research. We've been successful with both of those and continue to be so. From the research charities, I won't list them all, but particularly I think we should acknowledge the support and our successes with the Wellcome Trust, for example, and with industry as well. And I think sometimes we forget that biochemistry and bioscience generally doesn't just translate into the clinic and curing disease, but also into the bioscience economy as a whole in a broader sense. And so that's something we are keen to expand within the department, but also have a long tradition and a good presence at the moment, particularly for jointly supported graduate students between academia and industry. We're not all old and grey-haired. We've recruited a number of bright young things to lead new groups in the department. So amongst those is Bungo Akiyoshi, who we, I should choose my words carelessly, who we poached from the Dunn School of Pathology, who's just set up his laboratory on the basis of a Henry Dale Fellowship. Those are rather prestigious fellowships funded jointly by the Wellcome Trust and the Royal Society. With Andrew Angel, who we poached from East Anglia, who's on a Royal Society Fellowship. And then we have something new for biochemistry. We have a joint young group leader jointly appointed in the chemistry department to work on advanced proteomics, Shabazz Mohammed, and we lured him back from Utrecht. So we're managing to get people back across the channel at least. And we've got a number of other folk will join us at the end of the summer. So we're continuing to attract new people. One of the things we're particularly proud of, and I noticed purely by coincidence that somewhere else in this building there's an event celebrating women in science, and that's something we have quite a serious ongoing work to support more and more the work of women in science, and particularly in biochemistry, but we seem to be doing quite well in terms of some high-profile women in science. Alison is going to say a little bit more later on about the Royal Institution lectures at the end of last year. I'm sort of quickly looking around the room, but Elspeth Garland, it being the International Year of Crystallography, almost every week Elspeth comes and tells me of another radio program she's going to be explaining crystallography on, so that's going very well. And some of you may have noticed in the photos of new folk at the Royal Society. As you came in, that Judy Armitage was elected to the Fellow of the Royal Society earlier this year as well. So I think we're doing our bit, but there's clearly a lot more work to be done in that area, and it's something we take very seriously. And in terms of our research, it's intimately linked to our mission as a basic science teaching department. And both at the graduate level and the undergraduate level, that feeds into and enlivens and develops our research. And that's one of the reasons that we thought, particularly in terms of fundraising, that we would focus on support for graduate students and support for summer internships for undergraduates wishing to get a taste of the sharp end of research. So my profuse thanks to those of you who have already been able to support the department in this way, and my enthusiastic encouragement to any of you that are thinking of doing that in the future. It really does make a difference to supporting students going on into science. And that leads on a little bit to, we have a couple of our current second years here, Glenn and Philip, who are representing a new venture, which is the Oxford's entry to the iGEM competition, and they will tell you a little bit more about that, but they're, I think, a wonderful example of undergraduates, in this case, spearheading our new developments in synthetic biology, which Judy Armitage is also very much involved in, and Tony Watts has been also very much involved in setting up this iGEM project. I'm assured by Judy and Tony and others and our funding bodies that synthetic biology is going to transform our lives, and I certainly hope it does, because I'm reaching the age where I'm starting to think about a pension, and we need some enormous transformational industry to pay for my pension. So no pressure there, guys, but we'll hear a little bit about that later on. But one of the key things about this evening is to welcome you all back as alumni of the department, and so I'm delighted to introduce Professor Sue Harkley, who was formerly in biochemistry, was formerly at St Hugh's, and now is at, do I say Yesi or Yes-I?
Yesi.
Yesi, which is the York Environmental Sustainability Institute. And what I think is particularly exciting about hearing a little bit about Sue's trajectory is just to see how diverse the range of things that one can go on to following a basic education in biochemistry. And I think those of us who've been around a little while know that you can do lots of things with this. And I think we're going to hear one of the great things this evening. So I will shut up now and hand over to Sue. Thank you.
Thank you very much. It's great to be here and have an opportunity to tell you in the next 15 minutes or so how I got from a St Hugh's undergraduate in biochemistry to be director of the York Environmental Sustainability Institute. A bit of a mouthful, especially after the copious amounts of wine that are flowing, so that's why we say Yesi. And I want to talk about how I've sort of sustained that career, grown, and you can see it's been very hard work. This is my idea of teaching on a Tropical Biology Association course for African students in Uganda. So I want to spend a few moments thinking about how you can get from here to here. This is on the Christmas lectures, and this is a demonstration of just how much hot air comes out of university academics and cattle. And I wonder how, when my mother was asked to explain how I'd got from here to here and sort of summarise the achievements of the intervening 30 years in one pithy sentence to a family friend she hadn't seen for many years. She thought for a moment and said, she's put on a bit of weight. And I have done, that's just a warning to make sure you don't try and get in front of me in the canapé queue, but I have done a little bit more than comfort eating over the last 30 years, and it all started though as an undergraduate in biochemistry. And I got into key skills early on. Diligent study, good laboratory practice, just pointing me out because I've changed so much. And best of all, making sure that you get your fair share of the academic spoils. So, how did I go from undergraduate to Christmas lecturer? Well, you'll notice one consistent feature. You stand in the middle, touching the booze. Nothing strange. And I suppose the big change for me was at the end of my degree in biochemistry, I'd really enjoyed research, but I wanted to go and do biochemistry in what I thought was the real world. So I thought I wanted to do ecology. So I wrote to John Lawton, now Sir John Lawton, and said, I'm a biochemist, but I'd like to be an ecologist. Do you want a biochemist? Because he was an ecologist. And he wrote back and said, well, funny enough, I do. So I went off to do a PhD in ecology. And that led to all sorts of things. Botanical projects in Sinai, tropical forest regeneration in Borneo, and chasing boars in Tuscany. I still spend a lot of my time chasing boars, actually, just different kinds of ones. But what I noticed about the move to ecology was there were two good things. You could sit down on the job. Well, that's one good thing, actually. And then there was one bad thing, and that is you can only afford one pair of boots and 2 shirts. And I remember when I went to see the careers advisor, actually, in Oxford, and said I wanted to make this change. He said to me, what are you going to do then? Marry someone rich? Well, sadly, I didn't do that, but anyway. But what I did do was study how plants respond to herbivory. This is a bush cricket from Uganda, and you can see the chemical changes are very obvious when herbivores attack leaves, and it's that I spent my PhD studying. I got as obsessed as probably more healthy than. Probably more obsessed than is healthy with the phenylalanine pathway. And then, so even doing my PhD in ecology, biochemistry was really integral to what I wanted to do. And I thought to be a real ecologist, though, I should join the British Ecological Society. That's our learned body. And 25 years after I joined, I am the president-elect of that society, so I feel like a proper ecologist. And this is me introducing the International Congress of Ecology last year. And this was one of the tweets on the day. Sue Hartley starting the day in fine style. Now, my friends, some of you here tonight, who remember me as an undergraduate, will find that comment rather startling, because starting the day in fine style was definitely not what I did. And then I discovered who actually tweeted this, and it turned out to be one of my own PhD students, so I still suspect more than a hint of sarcasm. So I spent a career really studying the responses of plants to herbivory and the chemical mechanisms that mediate interactions between plants and their herbivores. This is my favourite herbivore, and it doesn't look like much, but this is a vine weevil, and it's one of the most severe pests around the world. It can attack over 400 different plants. And the great thing about it, in terms of women in science, is this is a parthenogenetic species. There are no males. So there we go. And one of the things I discovered early on was why the world is green, and that was before I discovered chlorophyll. But basically what we were talking about here was that plants have very many defences against the herbivores that eat them. And we've got a little bit more sophisticated recently, and we now understand a lot more about what plant secondary metabolites can do from the genetic level to mediating global climate change processes. So after I'd worked a lot on insects, I thought I would try larger, more intelligent herbivores. So I was working on sheep grazing in northeast Scotland, and I discovered two important things about fieldwork. Firstly, when you're building a tower to put a video camera on the top, make sure the final section is the right way up. Otherwise you'll be 30 foot above the ground in a howling northeastern gale and discover you can't get your feet in the steps properly because you've assembled it upside down. And the sheep were no more intelligent than I was, actually. This is an experiment looking at how sheep select their forage. So some of these patches of heather are fertilised and have high quality, and some have low. And the sheep are videoed and individually marked. So if anybody's struggling with insomnia, probably not what I'm talking, but if you are, there's plenty of sheep videos. Some you off. But one thing we noticed straight away was when sheep offered a choice of good quality heather or poor quality heather, went to these, the margarine tub lids we used to mark the plots up and ate those. So I abandoned sheep and worked on deer, and this is a different biochemical pathway. Their feeding behaviour is very much driven by monoterpenes present in pine needles. So you can see that chemistry is still at the root of things. And then I went to study climate change. So initially in greenhouses, elevated CO2 in these greenhouses, looking at how climate change would impact on the interactions between plants and their herbivores. And I'm particularly proud of my 1970s dress sense of fluorescent shorts, socks and hush puppies, which I thought was rather natty. And one of the things we were looking at was how changes in climate would shift the populations of herbivores and how they could move from one kind of plant to another. And we were looking at this work in Orkney, off North Scotland, and we were staying in a B&B with a journalist from The Scotsman. And he asked me what I was doing, and I said I was here to study new genetic variants of winter moth and the damage that they might do. And the next day, the headline in The Scotsman was, "Super moth could strip Orkney bare." So that was my first encounter with the media. And I'm still interested in climate change. I've just been awarded a visiting professorship at the Hawkesbury Institute in Australia, Sydney, Australia. And this is how to do things properly. This is a eucalyptus forest, and these are a free air enrichment facility. So this pumps out CO2. And so it's a kind of almost a natural system, but as if we were 100 years in the future, and we can see what the impacts will be. So after a spell in Scotland, I moved down south in search of better weather to Brighton, back to academia. The joys of academia, and particularly the joys of celebrating with students at the end of their exams. Every picture I have of myself, I'm clutching a glass of wine. And I started work on a new kind of defence in grasses, silicon. Grasses take up silicon, deposit it as hard granules, and cute little herbivores like voles don't like that very much. But we're quite mean to our voles, and we set up a big field experiment to see if we could demonstrate how plants would respond to a population explosion of voles. A bit like lemmings, you get these big peaks. So we put our voles in our enclosures, and what we discovered is that voles cannot actually climb horizontally. They climb up to here, because once they've eaten all the food and the plant is very well defended, they try and get out. They climb up to here, go along here, drop off, and climb up here, go along here, drop off. It's endless fun. So it's that kind of juvenile approach set me up very well for the Royal Institution Christmas lectures. I'm not going to say much more about this because we're going to hear from Alison, of course, but the topic was 300 million year war, which is, of course, the length of time that plants and animals have been battling it out. And chemistry is the medium of that battle. I wish my research group were this enthusiastic, I have to say. So, a bit of serious stuff just to finish. Back in York, 25 years after doing my PhD, as the founding director of the York Environmental, no, YESI, and we try and provide the evidence base for sustainable solutions to global environmental challenges. And after breakfast, so there's a lot to do, and we work on three main themes: sustainable environments, global change, and future food and fuel. They're all quite related. The big challenge, of course, is to be able to feed 9 billion people in a time of rapid social and environmental change. And there's a new urgency to my work on secondary metabolites and plant insect interactions now. This is the FAO predictions for crop yields with a 3-degree temperature rise in 2050. Red means a reduction in yield. One of the few places to see a likely increase in yield is North America. There really is no justice in the world. The people who cause the problem do not suffer from it. So this is very serious. Food shortages are linked to price rises and instability. And it's worse than that. Pest outbreaks are predicted to become a lot more common. And at the moment, this is our crop losses if we don't use pest control. Depending on our crop, almost between half and 3/4. If we do use pest control, that goes down, it's still high. But we are using, at the moment, chemicals to cause this change. To protect our crops, we use chemicals. It's not sustainable. And a lot of them are off the market because they're unsafe. And a lot of them have resistance. So we can't continue with that. And we need to find something to do because our crops have been disarmed by domestication. I don't know how many of you have seen ancestral maize. This is what maize used to look like. This is what maize looks like now. Bigger, better and juicier, but with no defences. So if you look at 203 different crops and what happens during their domestication, the biggest change, 60% change on average, is the loss of secondary metabolites that affect colour, flavour and toxicity. It's been the biggest change over domestication and it's been the most rapid and it's getting more rapid. Here's an example. These are ancestral potatoes from the Andes, full of secondary metabolites and resistant to blight. These are potatoes from Sainsbury's, look very nice, but completely susceptible to pests and diseases. So a big part of my work at the moment is trying to protect potatoes from their numerous pests and diseases. And it's a bit of a tough struggle. But one thing we can do is restore plants' natural defences. This delightful creature is called rootworm. Its larvae attack the roots of maize. But maize isn't defenceless. It releases a chemical, terpene, caryophyllene, and that attracts entomopathogenic nematodes. And I'm sorry to put this up just before we eat canapes, but these are the nematodes. That's not life size. These are the nematodes. They eat the larvae from the inside out and then are sprayed out afterwards. Now the interesting thing is ancestral maize has this chemical, it produces it, but modern maize does not. Well, at least modern American maize does not, but interestingly modern European maize does. So there you go. We're better defended than the Americans, at least in terms of maize. And I just want to end, because I'm not talking on, with a project that we've just started. It's funded by the Gates Foundation and BBSRC, and it's part of this initiative to increase global crop production. And our project is using wild ancestors to make rice more resilient to increasingly unpredictable water availability. The biggest crisis in world farming at the moment is water supply. Agriculture uses 70% of the world's available fresh water already, and huge fluctuations in floods and droughts is very difficult for farmers to cope with. And our crops are not as resilient to that as they used to be. This is cultivated rice. It provides the calories for half the world's population, so it's a vital crop. This almost completely unrecognisable. This is wild rice, ancestral rice. It looks more like a tree, it's perennial, and it's very resilient to all sorts of things. And this has 10,000 more genes than this. We've bred them out. We've bred out all sorts of things. We've bred out resistance to pests, we've bred out resistance to disease, and we've bred out resistance to drought. So we're trying to put it back in. Collaborators at Cornell are creating these chromosome substituted lines from donor parents, it's this, and breeding those segments back into the recurrent parent, that's elite cultivars. And we can then look, it doesn't look as neat as this actually, but anyway, it's a schematic, and we can look at which of those segments of genome make a difference to the resistant to osmotic stress. So the idea is to find these lines that maintain their yield under drought and to understand from transcriptomics and metabolomics how they do that. I think it's one of the most important projects I've ever been involved in, and it's hugely exciting. And I flew to India earlier this year, twice, not very sustainably, I have to say, but these are those lines in the field, under trial, in northeast India. And this is the Indian physiologist who's trialling them, and the early results are really promising. So, at last I'm doing something that I think could make a difference to a substantial number of people who are really on the edge of food security issues. So that's a bit of a serious end to what I'm about, but I have to say that most of my career has been anything other than serious, really. As part of my career, I've tried to get donkeys into lifts at the Royal Institution, everywhere with children and animals. I've been chased by chimps in the jungle in Uganda, and of course I was at the back because all these young things were miles ahead, and I could just hear one of the students shouting as we fled in terror. These huge chimps, you can see where humans get their aggression from, they're absolutely appalling. These big male chimps. We were running, I was at the back, and I could just hear the student shouting, "Don't look back!" Cheers, mate. And this is me studying Borneo tropical forest regeneration. It's 36 degrees, the humidity is 100%. When you're walking along, you can hear drip, drip, drip with the sweat coming off you. And he is wearing, I don't know if you can see it, an enormous furry hat with antlers on. It's one of these Christmas jolly hats. I don't know how his head didn't boil. Sinai Desert. We discovered a new species of tree in the Sinai Desert, as you do, since it's so little surveyed, and driving across the Australian outback. So it's been a fantastic 30 years, and an awful lot has changed from that bright young thing to this slightly more haggard and a little bit fatter, thank you, Mum, person. But one thing that hasn't changed is the friendship and support and fun I've had with my friends and colleagues that I met when I was at St Hugh's all those years ago, and they're here tonight, and that's the best thing of all. Thank you very much.
Well, thanks, Sue, that's great. And I'm really envious because the travel looks much more exciting than what I get. I should have switched to ecology.
Get out the lab, get out there, guys.
Get out the office. Okay. So we've had a sort of taster of Christmas lectures, and we've also heard about little worms. And that, without any pre-preparation, I think links us very nicely to Alison, who's going to tell us a little bit about what it actually takes to put together these Royal Institution lectures. So, Alison.
You need to switch these over.
Oh, sorry.
Sorry. It's great to see so many of you here. I hope you don't mind standing. You can sit on the floor if you want. Don't feel that you mustn't do that. So I want to start really by saying something about why I think it's a good idea for scientists to do public engagement. And I think there are many reasons for this. I think it's important that we justify the spending of public money and charity money. And I think it's important that we inspire the next generation. But one of the most important things, I think, is to make sure that science is properly entrenched in our society. That's something that's worth doing and something that's important to do. I don't know if you remember Sarah Palin. She ran for vice president in 2008, and she was governor of Alaska. And this is one of her really serious quotes. Dollars go to projects that have little or nothing to do with the public good. Things like fruit fly research in Paris, France. I kid you not. That was her real view. And that may mean that Sarah Palin is very stupid. Possibly quite bad. But it may also mean that maybe sometimes the message doesn't get out there properly about why we do the things that we do in the lab and what public benefit that might have. So not just in terms of medical research, but also in terms of intellectual nourishment and making science at the forefront of our cultural values. So I think that's a good reason to do some public engagement of science. And so this next slide is actually a picture of Charles Darwin's house in Kent, Down House. I have to confess I'm a bit of a Darwin bore. And I'm very interested in the history of Darwinism. And every year I take a group of undergraduates who have just finished their exams from Hertford College to Down House. We kind of have a very nerdy look round all the special exhibits and Darwin's collection cabinet and so on. And it was at this trip this time last year that I heard finally that I had been offered the job, if you call it a job, of giving the Royal Institution Christmas lectures. And it had a slightly strange history. So I got an email through in about February or March saying that I had been nominated as a possible Christmas lecturer, and would I like to put together a proposal for a narrative over 3 lectures on the subject of life. And I immediately deleted the email, as you would, thinking that sounds like a terrible thing to do. I couldn't imagine anything worse, I haven't got time, blah, blah, blah. And then nothing happened, and then a couple of weeks later I had another email from the Royal Institution saying, that's odd, most people will reply. Immediately, being very excited that they had been nominated for such a thing, and did you get the original email? And then I felt really embarrassed, and oh no, I did get the original email, so I dug it out and I thought about it, and I thought, well yeah, maybe I would put together something. Just because I had some ideas, and I thought it would get it out of my system at least. So I wrote a short kind of 3 pages on what I might put into a series of 3 lectures on life, and I sent it off, and I thought, well, I didn't hear anything more about it. And then I did hear more about it because they emailed me back and said that they wanted to do an audition. And I was like, an audition? Are you mad? I've never had an audition for anything. I even refused to go into a ballet show once because there was this thing about having to have an audition. So I was quite scared by that, and I confessed to my lab that this had happened, and they wanted to do an audition, and my lab thought it was absolutely hilarious. They just fell about laughing and said that I had to do it. So this guy came with a camera, wasn't too scary. I was thinking of a kind of Hollywood-style audition, but it was much more low-key than that. And I sat by my microscope and I talked about green fluorescent worms, like you do. Well, like I do, actually, nearly all the time. And it went from there. And it was at Down House this time last year that my mobile rang, and it was Ollie Brown, who I'm sure that Sue probably remembers from the RI, saying, "We finally decided that it's going to be you." And I said, "Oh, well, that's nice. Can I have a little bit of time to think about it, please?" And you're like, "No." So that was quite an interesting process. But having decided that this would be something worth doing and something I was keen and committed to doing, I have to say I'm very glad that I did, because it was an extremely enjoyable experience. So I just wanted to share with you a few insights into how you go about putting together something that ends up as a TV programme on science, and a few of the kind of funny things that happened on the way. And where it's got me to, what I'm going to do next, and what it might lead to in the future. So this is really to say that any kind of communication about science, whether it's a paper or a grant application or a seminar or a piece of public engagement, is all about storytelling. You have to tell a story that takes people with you. And actually, biology has the most powerful stories to tell. I think it's extraordinary. You hear any story, like the story that Sue's just told us about food security is an incredibly powerful story. So any of us who do any kind of biology, biochemistry, biophysics are surrounded by these very, very powerful stories and ideas that really are worth sharing. And any kind of story has a narrative arc. And telling a story is all about getting that narrative right. And so every story has a beginning and a middle and an end. And that was the way I went about putting together a series of 3 lectures on life. We called it Life Fantastic. And I'm a developmental biologist, so my beginning is quite an obvious one, really. This is a human egg that's just been fertilised by sperm. So this is where we all come from. This is a single cell. And from that single cell is derived all the 40 trillion cells or so, give or take a few million, that make up one of us. And as a developmental biologist, that's what gets me out of bed in the morning. That's the idea of trying to understand how all these cells are generated. So cells are the building blocks of all of us. How are they generated? How do they know what to do? How do they know where to go and what to do and when to stop doing that? And how does all that work? That's my burning question. And if you're going to tell a story, you have to have your burning thing that you are absolutely obsessed with and that you want to share with other people. And my way of putting this kind of story over is to make a problem out of it. And the problem is that it's all well and good to say that we work as organisms because all of our cells do different things at different times, and that's how it all works. And you can look at very different cells, like heart cells and neurons, and you can see that they're very, very different from one another. But how? How does that work? Because all cells have exactly the same DNA. Right? So how is it that cells end up doing different things? And of course we know that the answer to that is that DNA is organised into genes, and genes can be switched on and off. And that's a really key concept that most non-scientists don't understand. So it's telling people that genes can be switched on and off, so you can make proteins or not make proteins. And that's how cells become different from one another, right? Your heart cells will switch on heart genes, and your liver cells will switch on liver genes and switch off heart genes. And so that kind of regulation of gene expression is really key in developmental biology. But it's really key in one other extremely important area of biology as well, and that's evolution. And so my idea was to link together developmental biology and evolution, because the changes that make cells, the changes in gene expression that make cells distinct from one another during development are the same in molecular terms as the changes that make organisms different from one another during evolution, because gene expression patterns can change. So this is a really nice example that we use in the Christmas lectures. There's two types of stickleback. The top ones are marine sticklebacks and the bottom ones are freshwater sticklebacks. And the marine sticklebacks have these very well-developed pelvic fins. They have a kind of armour plating that they use for defence. And that's because in the marine environment there's lots of very nasty predators. Whereas the freshwater variety don't have this, because they don't need it. Because the gene that is responsible for causing the development of this armour plating is not expressed in these cells that would give rise to these pelvic fins, these very highly developed pelvic spikes in the freshwater fish. So there's a mutation, one mutation, that makes this kind of fish into this kind of fish. And it's a mutation in the regulatory region of a gene called PITX1, a transcription factor that controls the expression of other genes. So this is a very powerful way of saying that changes in gene expression are not only responsible for the development, but they're also responsible for evolution.